So, welcome to part 3 of the chapter 12 lecture on the nervous system. This is the last part, so we're almost done. We're going to start today by talking about signal transmission at synapses between nerves. So, a synapse is a region where communication occurs between two neurons or between a neuron and some sort of effector cell. The term presynaptic neuron, pre meaning before, is going to refer to a nerve that carries a nerve impulse towards a synapse. So in other words, it's the cell that's sending the signal. And then a postsynaptic cell is the cell that's receiving that signal. So it can be a nerve cell that would be called a postsynaptic neuron, post meaning after, that's going to carry a nerve impulse away from that synapse, or it can be some sort of effector cell that's going to respond to the impulse at that synapse. So there are a couple different options for synapses between neurons, and they can be one of 3 different types of synapses. We have axodendritic synapses. These are a synapse between an axon and a dendrite. We have axosomatic synapses. Would be a synapse between an axon and a cell body. And then we have axonic synapses, would be a synapse between an axon and another axon. So a synapse is going to be a junction between a neuron, between 2 neurons, or between a neuron and some effector. We've already said that. There are 2 different types of synapses that can occur. We have electrical or chemical synapses. In an electrical synapse, gap junctions are going to connect the cells and allow for the transfer of information to synchronize the activity of a group of cells. In chemical synapses, this is going to allow for one-way transfer of information from a presynaptic neuron to a postsynaptic neuron, or a postsynaptic cell. In electrical synapses, an action potential is going to conduct directly between the plasma membranes of adjacent neurons through gap junctions. And each gap junction is going to contain hundreds or 100 or so of these tubular connections, which are going to act like little tunnels to connect the cytosol between those 2 cells directly. So it's a direct connection between these 2 cells. So as ions flow from one cell to the next through these little connexons, the action potential is going to spread from cell to cell. There are 2 big benefits for electrical synapses. And the first is going to be faster communication. So because action potentials are going to conduct directly through those gap junctions, electrical synapses are going to be faster than chemical synapses. So at an electrical synapse, the action potential is going to pass directly from the presynaptic cell to the postsynaptic cell. The other big benefit of electrical synapses is synchronization. Electrical synapses can be synchronized or coordinated, and they can coordinate the activity of a group of neurons or muscle fibers. So a large number of neurons or muscle fibers can produce action potentials in unison if they are connected by gap junctions in these electrical synapses. The other type of synapse is a chemical synapse. So in general, plasma membranes of synapses are close, but they're not going to touch. They're separated by the synaptic cleft here that you can see in the middle here. So in a chemical synapse, the presynaptic neuron is going to convert electrical signals, the nerve impulse, into chemical signals, i.e. releasing neurotransmitters. And the postsynaptic neuron is going to receive the chemical signals, and in turn is then going to generate an electrical signal. That's our postsynaptic potential. So the time required for these processes at the chemical synapse to occur is called a synaptic delay, and it's going to be about half a millisecond. And this is going to be the reason that the chemical synapse is going to be a little bit more slow than our electrical synapse. So let's look at the steps that are involved in signal transduction across a chemical synapse. So first, a nerve impulse is going to arrive at our synaptic end bulb of our presynaptic axon, which is this guy up here. So our second step is then going to be the depolarizing phase of the nerve impulse is going to result in the opening of voltage-gated calcium channels on the synaptic end bulb. And because calcium ions are more concentrated in the extracellular fluid, that those calcium ions are going to flow inward through those open channels into that neuron. So this increase in calcium concentration inside our presynaptic neuron is going to serve as a trigger or a signal that's going to trigger exocytosis of our little synaptic vesicles. And these synaptic vesicles are going to be packed to the brim with neurotransmitters. So those neurotransmitters are going to, or those synaptic vesicles are going to bind to the cell membrane, and those neurotransmitters are going to be exocytosed into that synaptic cleft. Then the neurotransmitter molecules are going to diffuse across the synaptic cleft, and they're going to bind to the neurotransmitter receptors on the postsynaptic neuron's plasma membrane. All right, once the binding of those neurotransmitter molecules has occurred to their little receptors, this is going to trigger a ligand-gated channels to open, and the channel is going to allow a very particular type of ion to flow across the membrane. And the type of ion that's flowing across the membrane is going to depend on the type of neurotransmitter that's being released. And this is going to influence whether or not we have an excitatory or an inhibitory postsynaptic potential. So as those ions flow through that opened channel, the voltage across the membrane is going to change, and this change in membrane voltage is going to be called the postsynaptic potential. So depending on which ions the channel allows entry, the postsynaptic potential may be depolarizing in that it's going to be excitatory. It's going to cause the production of another action potential, or it can be hyperpolarizing, where it's inhibitory and it prevents an action potential from forming. And then our last step is when a depolarizing postsynaptic potential reaches threshold, it's going to trigger an action potential in the axon of that postsynaptic neuron. So let's talk about these postsynaptic potentials. There are two different possibilities for these. We can either have an excitatory postsynaptic potential, an EPSP for brevity's sake, and this is going to be a depolarizing postsynaptic potential, and it's going to be caused by the opening of sodium channels. Alternatively, we can have an inhibitory postsynaptic potential, an IPSP. This is a hyperpolarizing postsynaptic potential, and it's caused by the opening of chloride or potassium channels. So a postsynaptic neuron can receive many different signals all at once, but whether or not an IPSP or an EPSP is going to occur is going to depend on which of the stimuli is greatest. So as far as structures of neurotransmitter receptors are concerned, there's a couple different options that these neurotransmitters combine to elicit their response. So neurotransmitters are at these chemical synapses are going to cause either an excitatory or inhibitory graded potential. And neurotransmitter receptors can have two possible structures. We've got ionotropic receptors, where a receptor is going to contain it's going to have a receptor for a neurotransmitter binding site as well as an ion channel. And then we have metabotropic receptors. These are receptors that contain a neurotransmitter binding site but no ion channel. So looking a little bit more closely at our ionotropic receptors, these, like we mentioned, are receptors that contain a neurotransmitter binding site and an ion channel. So we've got two different examples of these ionotropic receptors. The first one is an ionotropic acetylcholine receptor, which you can see up here at the top. This is going to contain two binding sites for our neurotransmitters as well as a cation channel. So when we have binding of our acetylcholine to these two receptors, it's going to cause a cation channel to open, and it's going to allow for the passage of the three most plentiful cations to enter the cell. In this case, it's sodium, potassium, and calcium. When this occurs, an EPSP is going to be generated. Alternatively, then, we have ionotropic gamma aminobutyric acid, or GABA, receptors. It's going to contain two binding sites for our neurotransmitter GABA, again, as well as a chloride channel. So when we have our little GABA neurotransmitter molecules bind to those receptors, it's going to cause our chloride ion channel to open, and it's going to allow for this big influx of chloride ions into the cell, and this is going to cause an IPSP. So it's going to be an inhibitory postsynaptic potential. Then we have the metabotropic type of receptors. Again, these are receptors that contain a neurotransmitter binding site, but they do not have that ion channel attached to it. So these are receptors. These receptors are going to be coupled to a separate ion channel by a type of membrane protein called a G protein. So these are G protein coupled receptors. They're coupled with this G protein. So when our neurotransmitter binds to our metabotropic receptors, it's going to cause our G protein to directly open our ion channel or close, depending on the case. Or it can have, or when our neurotransmitter binds, that G protein can use a secondary messenger, something in the cytosol that's going to trigger the opening or closing of that ion channel. So this G protein can either directly or indirectly open that ion channel. So it can, Once we've had enough of whatever influx or efflux of ions, we need to sort of get rid or dispel these neurotransmitters. So they need to be removed from the synaptic cleft. And this happens in one of three ways. They can either just diffuse out of the synaptic cleft. Some sort of enzyme can come in and degrade that enzyme, break it down so that it's not going to bind anymore. Or it can be taken back up into the cell to be reused later. So those are our three options for how we kind of clear out our neurotransmitters once we've elicited the appropriate response. So if the presynaptic end bulb releases their neurotransmitters at the same time, the combined effect can generate a nerve impulse, and this is due to summation. So summation can be either temporal or spatial. So in spatial summation, this is summation of a postsynaptic potential in response to stimuli that occurs at different locations in the membrane of the postsynaptic cell at the same time. All right, so we've got multiple different locations that are giving off the stimuli. All right, so for example, spatial summation is going to result from the buildup of neurotransmitters being released by several presynaptic end buds. All right, alternatively, then, we have our temporal summation, and this is summation of a postsynaptic potential in response to stimuli that occurs at the same location in the membrane of that postsynaptic cell, but at different times. So, for example, the temporal summation results from a buildup of neurotransmitters being released by a single presynaptic end bulb, happening two or more times in rapid succession. So if you think about this in terms of people, if in order to have our action potential sparked up or initiated, we all have to hit enter. So in spatial summation, that would be if everybody in the class hit enter to initiate that action potential. All right, multiple different people hitting enter to release that neurotransmitter, so that we have summation there. That would be spatial summation if everybody hits enter all together. All right, temporal summation would be if I myself just hit enter a bunch of times repeatedly in rapid succession. It only takes one of me to hit enter a bunch of times. That's temporal summation. Whereas in spatial summation, we all have to hit enter for that summation to occur. So, yeah. So, summation of postsynaptic potentials can have three main impacts. So, a single postsynaptic neuron is going to receive input from several different of these presynaptic neurons, some of which are going to release excitatory neurotransmitters, others are going to release inhibitory neurotransmitters, and the sum of all of the excitatory and inhibitory effects at any given time is going to determine the effect on the postsynaptic neuron, which can respond in one of three ways. All right, the first option is going to be for an EPSP to occur. So if the total excitatory effect is greater than the total inhibitory effect, but it's less than the threshold level of stimulation, then the result is going to be an EPSP that does not reach threshold. All right, so it's close. It's almost excitatory, but it doesn't quite make the cut. So following an EPSP, subsequent stimuli can more easily generate a nerve impulse through summation because the neuron is already partially depolarized. Our second option is going to be that a nerve impulse occurs. So if the total excitatory effect is greater than the total inhibitory effect and threshold is reached, then one or more nerve impulse or action potentials will be triggered. These impulses are going to continue to be generated as long as the EPSP is at or above the threshold level. And then our final option for what can occur is an IPSP. So if the total inhibitory effects are greater than the excitatory effects, then the membrane is going to be hyperpolarized. It's going to have an IPSP, an inhibitory postsynaptic potential. And the result of this inhibition of that postsynaptic potential and an inability to generate a nerve impulse. All right. So here is just a lovely little table looking or illustrating the various parts of the neuron. Kind of oddly placed, but it's there. So next we need to discuss the various neurotransmitters that are used within the body and how they work. So neurotransmitters are broken up into all different kinds of categories. A big one, a very popular one, is going to be small molecule neurotransmitters. This is going to include things like acetylcholine, amino acids, biogenic amines, ATP and other purines, as well as nitric oxide and carbon monoxide. All right. So acetylcholine is the first one, and this is a big one because it is the best studied neurotransmitter known to man. Everybody loves acetylcholine because we know a lot about it. This is released by many of the different PNS neurons as well as some of the CNS neurons. So acetylcholine is an excitatory neurotransmitter at some synapses, such as at neuromuscular junctions, where the binding of that acetylcholine to an ionotropic receptor is going to open cation channels. But it can also be an inhibitory neurotransmitter at other synapses. So this would be an instance when it would bind to a metabotropic receptor that's coupled to a G protein receptor, that's coupled to a G protein that would result in the opening of a potassium channel. Our next group of neurotransmitters in the small molecule neurotransmitter category would be amino acids. So several of the amino acids that are in the body do act as neurotransmitters in the central nervous system. So things like glutamate is going to have a powerful excitatory effect. So many excitatory neurons in the central nervous system, and about half of the synapses in the brain are going to communicate via glutamate production or secretions. So at some glutamate synapses, binding of the neurotransmitter to ionotropic receptors is going to open cation channels. And the consequence is going to be an influx of cations that's going to produce an EPSP. Another amino acid would be gamma aminobutyric acid, or GABA. This is an important inhibitory amino acid neurotransmitter. So at many synapses, the binding of GABA to an ionotropic receptor is going to open a chloride channel. And GABA is found only in the central nervous system, where it is most commonly an inhibitory neurotransmitter. So anti-anxiety drugs like diazepam, commonly called brand name Valium, are going to enhance the actions of GABA. So our next group of small molecule neurotransmitters are called biogenic amines. So certain amino acids can be modified and decarboxylated to produce these biogenic amines. Right? And there are a couple that are really important. So norepinephrine is one, and it's going to play a major role in arousal, like awakening from sleep, like waking up, as well as dreaming and the regulating of mood. And then a smaller number of neurons in the brain are also going to use something called epinephrine as a neurotransmitter. And this one also serves as a hormone in other places in the body. So brain neurons containing neurotransmitter called dopamine are going to be active during emotional responses, as well as in addictive behavior and pleasurable experiences. So in addition, dopamine releasing neurons can help to regulate skeletal muscle tone and some aspects of movement to contract, can also help regulate skeletal muscle tone and some aspects of movement due to contraction of skeletal muscles. So the muscular stiffness that you see in patients who have Parkinson's disease is a result of the degradation of these dopaminergic neurons in the brain. And if you actually look at brain scans or histology sections of patients with Parkinson's, you can actually see the degradation or the loss of these dopaminergic neurons, which is why they have that kind of weird stiff movement associated with the disease. So norepinephrine, dopamine, and epinephrine are all classified chemically as catecholamines. Right? Our next, just for a few more of these small molecule neurotransmitters, we have ATP and other purines. Remember, purines are those membered rings that we have that make up proteins. Sorry. So the characteristic ring structure of the adenosine portion of ATP is called a purine ring. So adenosine itself, as well as the triphosphate and diphosphate and monophosphate derivatives, ATP, ADP, and AMP, are going to work as an excitatory neurotransmitters in both the central nervous system and the peripheral nervous system. We also have something called nitric oxide, and this is an important excitatory neurotransmitter that's going to be secreted in the brain, the spinal cord, the adrenal glands, as well as in the nerves of the penis, and has a widespread effect throughout the body. So that's an important one. And then last but not least, we have our carbon monoxide. This is a gas, and it's also an excitatory neurotransmitter that's produced in the brain as well as in response to neuromuscular and neuroglandular functions. So carbon monoxide might help to protect against excess excess neuronal activity, and it might be related to the dilation of blood vessels, as well as memory, olfaction, vision, thermoregulation, also insulin release, and anti-inflammatory activities. So it does a lot, but we're still kind of studying this one on how exactly it works. Obviously, too much carbon monoxide is very poisonous. Our next group of neurotransmitters are called neuropeptides. We're not really going to get in too much to these. There's a table right here for you if you're interested in learning more about them. You can read up on them, but I'm not going to go into them in any great detail for this particular lecture. So let's talk about neural circuits now. We've got a couple different options for these. So a neural circuit is a functional group of neurons that processes specific types of information. And there are multiple types of neural circuits. We've got simple series, diverging, converging, reverberating, and parallel after discharge neural circuits. So starting with, let's just mention the simple circuit. This is just basically what it sounds like. One neuron is going to stimulate is going to transmit that impulse to a second neuron, which on to a third neuron, on and on. It's simple. All right. Next, we then have the diverging circuits. All right. This is when a nerve impulse from a single presynaptic neuron causes the stimulation of an increasing number of cells in the circuit. So one neuron is going to stimulate two, which stimulates a few more, on and on. We have a diverging circuit occurring, a stimulation of an increasing number of circuits, of neurons. So for example, a small number of neurons in the brain that govern a particular body movement are going to stimulate a much larger number of neurons in the spinal cord. So sensory signals are also arranged in diverging circuits, and this allows a sensory impulse to be relayed to several different regions of the brain. And this arrangement is going to amplify the signal that's being sent. Next, we have our converging circuits. It's multiple neurons are converging on one neuron. So the postsynaptic neurons are going to receive, technical troubles. So these postsynaptic neurons are going to receive, our postsynaptic neuron is going to receive nerve impulses from several different sources. So for example, a single motor neuron that synapses with a skeletal muscle fiber is going, with skeletal muscle fibers at that neuromuscular junction is going to receive input from several pathways that are going to originate from different regions of the brain. Next, we have a reverberating circuit. So it starts off pretty simple. The incoming impulse is going to stimulate the first neuron, which stimulates the second neuron, which stimulates the third neuron, so on and so forth. And then branches of the later neurons are going to synapse with earlier neurons. All right, so they're synapsing back. All right, and this arrangement is going to send impulses back through the circuit again and again. So the output signal may last for a few seconds to many hours, depending on the number of synapses and the arrangement of the neurons in that circuit. So these are a little bit longer lasting. And then last but not least, we have our parallel after discharge circuits. This is when a single presynaptic cell is going to stimulate, a group of neurons, each of which synapses with a common postsynaptic cell. All right, so we've got our one long presynaptic cell that's stimulating a group of neurons here, each of which are going to synapse with a common postsynaptic cell down here. So we're sharing. And then a different number of synapses between the first and the last neuron are going to impose a varying synaptic delays, so that the last neuron that exhibits, is going to exhibit multiple EPSPs or IPSPs. So if the input is excitatory, that postsynaptic neuron then can send out a stream of impulses in quick succession. So parallel after discharge circuits may be involved in precise activities like mathematical calculations. So work on your parallel after discharge circuits when you're doing your math homework. And then last but not least, we need to talk about the regeneration and repair of nervous tissue. So although the nervous system exhibits plasticity, neurons are going to have a limited ability to regenerate themselves. So plasticity is going to be the capability to change based on experience, whereas regeneration is going to be the capacity or the capability to replicate or repair itself. So that brings us into neurogenesis in the central nervous system. In the CNS, there's not really going to be a whole lot of repair that occurs to any sort of damaged neurons. This is due partly because of the inhibitory influences from neuroglia, particularly the oligodendrocytes. Also, it's because this is caused by the absence of growth-stimulating cues that would be present during fetal development. And it's also going to be caused by a rapid formation of scar tissue. So scar tissue is going to form so rapidly that any kind of damage is not going to be able to be repaired. So when they say that your brain cells don't grow back, there's a little bit of truth in that. But anyhow. So that brings us then to the damage and repair of the peripheral nervous system. All right. So in the PNS, repair is going to be possible if the cell body is intact, if the Schwann cells are functional, and if scar tissue formation does not occur too rapidly. All right. So steps for this are going to involve the repair. Steps in this repair process are going to be chromatolysis, Wallerian degradation, and then the formation of a regeneration tube. So let's look at how this happens for the damage and repair of our peripheral nervous system. So about 24 to 48 hours after injury, those nyceal bodies are going to break up into fine granular masses. And this alteration, this change in those nyceal bodies is called chromatolysis. So by the third or to fifth day after the injury has occurred, or the damage has occurred, the part of the axon that's distal to the damaged region is going to become slightly swollen, and it's going to break up into fragments, like you can see here. The myelin sheath is also going to deteriorate, but the neurolemma is going to remain, and that's important. The degradation of the distal portion of the axon and the myelin sheath is going to then be called Wallerian degradation. So macrophages are going to phagocytize any sort of debris that's hanging around after this injury, and they're also, and then also the synthesis of RNA and protein is going to be really kicked up into gear, and this is going to favor rebuilding or regeneration of that axons. So we're kicking it up. Then the Schwann cells on either side of that injured site are going to multiply, or multiply via mitosis, and they're going to grow towards each other, and they may form this regeneration tube, which we see here. And this regeneration tube is going to span across the injured area. And this regeneration tube is going to help to guide the growth of new axons from the proximal area across the injured area into the distal area that was previously occupied by the original axon. So the new axons cannot grow if the gap at the site of injury is too large. So if it can't span that gap there, or if the gap becomes filled with collagen fibers, like and there's scar tissue that forms. So that would hinder any sort of repair to the peripheral nervous system nerves there. So those are our 3 steps: our chromatolysis, our Wallerian degradation, and then the formation of that regeneration tube. And that is how we repair damage to the peripheral nervous system. And that is the last of this lecture. So I will see you in class. If there are any questions, please feel free to email me or, talk in class if there are still questions.